it's important to understand life and how life works and how brain works. How all these things put together, which eventually lead to our minds and our brains. We are using a combination of microscopy, physiology, and animal behavior to study and understand how the sensory information is interacting with the internal dynamics of the brain and how these interactions lead to behavioral plasticity. So we are interested in these ancestral corticolimbic structures in zebrafish brain that correspond to our hippocampal and triangle-like areas. And we want to understand the universal circuit architecture and computations that underlie how these brain regions leads to learning and they provide us this behavioral plasticity. My lab have been working on habenula. This is a brain region that is important for predicting outcomes, which, as you can imagine, important for both learning but also for decision making. As animals developed, habenula also developed, but in a way where distinct functional modules of the circuit is added sequentially. For example, one module is born at a certain developmental stage and the next module born, and so on and so on. Interestingly, in a separate paper, we also show that actually as animal develops, they actually gain new abilities in their learning. For example, a really young zebrafish are not very good in learning, but as they develop, they actually get better and better in learning. Actually, when we perturb distinct modules of this circuit, we also find out that not only the learning can be perturbed, but also relearning can be perturbed. In a way, for example, animals learn one rule, but then during the experiment, we change the rules of the game, so they have to learn a new rule. And actually perturbing that particular nucleus in habenula lead to actually an impairment in this reversal learning, which was also, I think, at least to me, it was very exciting.